Written everyone, pathogenesis of atopic dermatitis. The pathogenesis of AD can be divided into three major categories. 1. Epidermal barrier dysfunction. 2. Immune dysregulation. And 3. Alteration of microbiome. These mechanisms interact with each other and are modulated by both genetic and environmental factors. Genetic factors account for a large proportion of susceptibility to early onset of AD, with a significantly higher concordant rate in monozygotic twins, 77% compared to dizygotic twins, 15%. Although the entity in the atopic triad cluster together families, a parental history of AD is a stronger risk factor for the development of AD than either asthma or allergic rhinitis, supporting the existence of genes specific to AD susceptibility. Genes that encode proteins important to the epidermal barrier and immunologic function have been implicated in AD pathogenesis. AD is a complex genetic disease, and both gene-to-gene and gene-to-environment interaction have important roles. Epidermal barrier dysfunction. A defective epidermal permeability barrier represents a consistent feature of AD and is evident in non-lesional as well as lesional skin of affected individuals. Manifestation of epidermal barrier dysfunction include increases in transepidermal water loss, pH, permeability, as well as outer lipid composition. The level of transepidermal water loss in non-lesional skin of children with AD correlates with disease severity. Epidermal barrier dysfunction permits an easier entry for irritant, allergen, and microbes, which trigger immune response that include the release of pro-inflammatory cytokines. In infants, rectal transepidermal water loss is associated with an increased likelihood of epicutaneous sensitization to aero allergen, which could potentially play a role in development of asthma and allergic rhinoconjunctivitis. Factors that contribute to impaired cutaneous barrier in AD are discussed next. Filaggin and other structural proteins. Filaggin is a keratin filament aggregating protein that serves as a major structural component of stratum corneum. FLG loss of function variant represent the strongest known genetic risk factor for AD and are also responsible for ichthyosis vulgaris, with carrier frequencies of up to 10% in European and 3% in East Asian population. Approximately 20 to 50% of European and Asian children with moderate to severe AD have at least one FLG loss of function variant. The penetration of AD is 40% for one and 90% for two loss of function allele. This implicated epidermal barrier dysfunction in the initiation of AD, with subsequent development of Th2 biased immune responses, although FHE loss of function variants also play a role in pathogenesis and persistence of AD in children of African descent. They appear to be rare individuals from sub-Saharan Africa. However, technical limitations may have reduced the detection of uncommon FLG variants in earlier studies. Of note, filaggin expression is also affected by intragenic copy number variation and reduced by increased local pH, protease activities, and Th2 cytokine levels. FLG mutations are associated with early onset AD, greater disease severity, and persistence into adulthood, as well as enhanced epicutaneous sensitization and increased risk of irritant contact dermatitis and eczema, herpes simplex virus infection, and food allergy. FLG mutations have also been linked to increased risk for the development of asthma and greater asthma severity. However, these effects are only seen in patients with pre-existing AD. Since filaggin is not found in the gastrointestinal or chronic mucosa. The association of LG mutation with food allergy and asthma strongly suggests that epicutaneous sensitization and/or cutaneous inflammation can contribute to the development of systemic atopic disease. Filarin breakdown products such as histidine contribute to epidermal hydration, acid metal formation, lipid processing, and barrier function. Gene expression profiling and immunohistochemical analysis of lesional and non-lesional skin from AD patients have shown broad effects in terminal differentiation with downregulation of other epidermal barrier proteins such as loricrin, corneodesosin, involucrin, small rich protein 3 and 4, claudin 1, and glyconified envelope protein 2b. Stratum corneum lipid. The composition, organization, and biochemical processing of stratum corneum lipid are critical determinants of epidermal permeability barrier function. In AD, filakin deficient cytoskeletal scaffold contribute to abnormal loading and secretion of lamellar body, with subsequent defects in post-circulatory lipid organization and processing. Disruption of skin acidic mantle leads to reduced activity of lipid processing enzymes such as beta-glucocerebrosidase and acid-sphingomyelinase. Th2 cytokine also negatively affect the generation of stratum corneum lipid component. Lastly, Staphylococcus aureus colonization of the skin affect lipid composition and contribute to epidermal barrier impairment. Proteases and protease inhibitors. Lesional AD skin demonstrate elevated level of endogenous serine protease, callitrain 5 and 7, due to imbalance in the activities of the proteolytic enzyme and protease inhibitor, such as lymphoepithelial calcium-type trypsin inhibitor, encoded by SPINK5 biallelic loss-of-function SPINK5 mutation underlying Netherton syndrome, which feature profoundly compromised barrier function and atrophy, while SPINK5 polymorphism has been linked to increased risk of AD in some populations. Other factors that enhance proteolysis include increased skin surface pH and exogenous proteases from allergens. How does my pollens, Staph aureus, and Malassea lactate deficiency results in excessive duration of the corneal-desmosomal component, desmolite 1, causing abnormal stratum corneum detachment and thereby disrupting the epidermal barrier. The Staph aureus extracellular Va protease have a sequence similar to those of Staph aureus exfoliative toxin and is also thought to degrade DHT1. In addition, unrestrained protease activity leads to degradation of lipid processing enzymes and antimicrobial peptides as well as activation of pro-inflammatory cytokines. Immune dysregulation. Both innate and adaptive immune systems play dramatic interrelated roles in the pathogenesis of AD. Inflammatory profile is diverse, with activation of skin resident, inflammatory dendritic cell, rupture innate lymphoid cell, and Langerhans cell. Acute AD lesion have a predominance of Th2 cytokines, but there is subsequent evolution to a chronic phase characterized by Th1 and Th22 cytokine profiles, as well as variable levels of Th17 cytokines in both acute and chronic AD. The acute phase features interleukin-4, interleukin-5, interleukin-13 activation of eosinophil and mast cell and production of allergen-specific IgE. Keratinocyte-derived cytokines include interleukin-1, submesomal lymphopoietin, interleukin-25, and interleukin-33 promote a Th2 immune response Th2 cytokine inhibits expression of major terminal differentiation proteins such as loricin, filakin, and involucrin, as well as beta-defensin 3 and 2, antimicrobial peptide.
Thymic-stromal lymphopoietin TSLP interleukin-7-like cytokine that is known as the master switch of allergic inflammation due to its central role in evoking a Th2 response via dendritic cell activation, exposure to allergens, viral infection, trauma, and other cytokines. Interleukin-1 beta TNF can trigger TSLP production by keratinocyte, fibroblasts, and mast cells. TSLP is highly expressed in acute and chronic lesions of AD, but not in non-lesional skin of patients with AD or in unaffected individuals. Interleukin-4 and interleukin-13. Interleukin-4 has a key role in driving Th2 cell differentiation, IgE production, and eosinophil recruitment. Transgenic mice overexpressing interleukin-4 in the epidermis develop atopic dermatitis-like lesions, pruritus, and altered microbiome, and elevated IgE levels. The heterodimeric receptors for interleukin-4 and interleukin-13 both contain interleukin-4 receptor alpha unit, an activate signal transducer and activator of transcriptase, which promote the differentiation of naive T cells into Th2 effector cells. Although interleukin-4 and interleukin-13 share a 25% sequence, homology, and effector function, studies in human subjects and human keratinocyte cell line support an independent role for interleukin-13 in AD pathogenesis. Monoclonal antibodies that target interleukin-4 or 13 represent effective treatment for AD. Other cytokines. Th17 cells are important in the regulation of innate immunity, in particular neutrophil recruitment, and have also been implicated in allergic disorders. Th17 cells are found in acute as well as chronic AD lesions, and production of interleukin-17 and interleukin-19 is especially characteristic of new-onset pediatric AD. Interleukin-26, which is also produced by Th17 cells, induce production of Th2 and Th17-associated cytokines, thereby potentially acting as a bridge between Th2 and Th17 responses. Interleukin-31 is a Th2 cytokine that is highly expressed in lesional skin and serum of patients with AD as well as other related skin disorders, such as prurigo nodularis, cutaneous exposure to staphylococcus superantigen rapidly induces interleukin-31 expression in atopic individuals, establishing a link between staphylococcal colonization of the skin and pruritus. The heterodimeric receptor for interleukin-31 is expressed in keratinocyte, eosinophil, activated macrophage, cutaneous nerve fiber, and dorsal ganglia. Randomizable control trials have shown that nemolizumab, a humanized monoclonal antibody against the interleukin-31 receptor A unit, can significantly reduce pruritus in patients with moderate to severe AD, although improvement in disease severity score were modest. Interleukin-33, a member of interleukin-1 cytokine family, protects against helminth infection by promoting Th2 type immune response. Interleukin-33 expression is increased in AD lesions compared to the skin of unaffected individuals, and AD therapies targeting interleukin-33 are underdeveloped. Innate lymphocells. The innate lymphocyte family include natural killer cells and three of non-cytotoxic ILC that orchestrate immunity, inflammation, and homeostasis in multiple tissues. The rupture ILC population expanded in AD lesion and stimulated by TSLP, interleukin-25, and interleukin-33. ILC2 interact with other immune cells, mast cell, eosinophil in the skin to promote Th2 type inflammation in a T-cell independent manner. The cutaneous microbiome. The cutaneous microbiome represents a complex and highly diverse community of pathogenic and commensal bacteria, fungi, viruses that play a critical role in epidermal homeostasis with microbial dysbiosis, contributing to the pathogenesis of AD. More than 90% of patients with AD have skin colonized with Staph aureus, compared to about 5% of uninfected individuals, presumably reflecting the disrupted acid mantle, decreased antimicrobial peptides, catalycidin, defensin, and anti-cytokine milieu of AD skin. During AD flare, bacterial diversity decreases. The proportion of microbiome counted for staphylococcal spp increases from 35% to 90%. Conversely, normalization of microbial population correlate with clinical improvement in AD. Superantigen can promote the development of a Th2 immune response. An exotoxin with superantigenic property and reduced by up to 65% of the Staph aureus strain that colonize AD patients, compared to uninfected control, and actually respond to the Staph aureus superantigens. Enterotoxin A and enterotoxin B occur more frequently in patients with AD. The Staph aureus delta toxin also stimulates mast cell degranulation and tissue inflammation. In addition to filaggin deficiency, increases the susceptibility of keratinocytes to Staph aureus, and alpha-toxin induces cytotoxicity. Lastly, malassezia spp may also contribute to inflammation in AD. And adults with severe head and neck disease often display IgE reactivity to malassezia antigens. Alteration in skin microbiome of AD patients related to the use of cleansers and topical immunomodulatory or antimicrobial agents may have potential effects on cutaneous inflammation and barrier function. Topical administration of coagula-negative staphylococcal strain with antimicrobial activity on the gram-negative commensal Rhodomonas mucosa have been so markedly reduced Staph aureus colonization in AD patients. Our mucosa application was also associated with decreased AD severity and topical corticosteroid requirement, providing the basis for bacterial therapy as a potential AD treatment.